Hello, everybody.
Hello.
Hello.
Hello, thank you so much for joining us. We're admitting people in and we will begin shortly. And
Hello. Good afternoon.
hello,
Hello. Good afternoon.
good afternoon. Please go ahead and put where you're joining us from in the chat. We'd love to know where you're connecting from, and we'll begin right away. Today, our speaker is Dr. Pascal Okwiri Ojwang, and Dr. Pascal is a senior lecturer in the Department of Crops, Horticulture and Soils at the Egerton University in Kenya. He has a background in plant breeding with interest in breeding tropical cereals and legumes for resistance to biotic, abiotic stress tolerance, and quality traits. He's a research leader and coordinator of graduate training research projects at Egerton University. Previously, he worked as dryland legumes breeder at Kenya Agricultural Research Institute, KARI, currently renamed as the Kenyan Agricultural and Livestock Research Organization, KALRO. He has published over 30 research journal articles in peer-reviewed international journals. And today he's going to be speaking to us about experimental design and data analysis in plant sciences. And at this point, I'll hand it over to you, Dr. Pascal, and we're very excited to have you here. Thank you so much for honoring our invitation again. So over to you.
Okay, good afternoon, and thank you, Olfunke, and the entire audience. I'll be giving a talk on experimental designs, basically the principles, the design aspects, and some questions regarding the choice of a good experimental design, and what is the use of modeling in research. So to begin, I'll look at some of the design terminology. Then I'll go into one way, ANOVA, that is just single factor or one factor experiment. I will go into details with that, and then the others I'll just do an overview because it will be a repeat of what I would have covered within this, because usually the analysis of experimental design follows a single pattern, and it's always easy to kind of explain using the one factor experiment. It's always simpler. So then I look briefly at the randomized complete block design. I look at the factorial treatment structure, split plot designs or experiments, and then some just short questions regarding a good design and the role of a statistical model. So just to begin with, okay, what is an experimental design? An experimental design is simply a procedure. For conducting research by looking into the objectives, but then you do it in a controlled manner. The whole idea is to maximize precision so that you can draw specific conclusions. Because before you start research, you usually have some research questions, and so you have an objective to answer to those research questions. So what is it all about? It is about looking at the effect of independent variable on a dependent variable. So then what is analysis of data? It is simply the whole process of collecting the data, you need to validate the data, then you test the assumptions of the analysis of variance. Before you run the data, you have to test whether it meets the assumptions of the analysis before you can go into modeling. Okay, so you model the raw data so that you can support the decision making. Now, the interpretation of the results must be insightful, and you must look at the trends within the data. That is what is important, so that you have objectivity in terms of the conclusions you draw for your reporting and sharing your data, sharing your results with the audience. Now, differences between your treatments should be based on statistical tests. So that is very important. Now, there are certain main principles of experimental design, and I just want to look at them. So what are the basic principles? So you have replication, you have randomization, and you have blocking, or you look at the covariates. Like sometimes you run your data, for example, disease attacks, and your experiment was not testing on the disease, so then it interferes with the results. So then you have to score for the disease as a covariate, so that now when you run the data, you have to remove the effect of the disease so that you have clear effect of your treatments. So usually replication is just for quantification of natural variation, and it allows you to estimate your error. Randomization is about removing the confounding effects between your data, and then the blocking, you reduce the experimental noise, particularly when you're dealing with field experiments, because you expect some gradient in terms of fertility or other field effects that may affect the quality of your results. Because when you don't consider that, then you have contamination in terms of the results that you present. So there are these terminologies that confuse people a lot. So what is a response? What is a factor? What is a factor level? What are treatments? And what are covariates? Those are some of the things that you have to be clear about. What you measure in the field or in a greenhouse where you're doing your experiment is your response. Then the categories of your factors, those are what you call the categories of the variables that you measure. Like, for example, if you have nitrogen, maybe you have variety, and any other factor, maybe irrigation, then those are your factors. Variety is a factor, nitrogen is a factor. Now, the combinations of those factors in the experiment are what you call the treatments. Then the covariate, like I explained, these are variables that you measure alone that may help you in terms of reducing the experimental error. So the unit is a plot for plant researchers like us. And then the measurement unit is where you take the measurement. And then replications, like I said, is just the repetition, so that all the replications receive the same treatment. But that is for a balanced design. You may have an unbalanced design. Then the blocks are just homogeneous units, like plots that look alike. Like in the field, if you have a gradient, they say the upper part of the field will be uniform, but it will vary because you expect the lower part to be drier or sorry, the higher part of the field will be drier and the lower part will be wet, you expect. And then in terms of fertility, also you expect the lower part of the field to be having higher fertility. So when you are testing, you must choose a design that will help you to partition your field into similar or homogeneous units so that you minimize bias. Now, usually we aim at randomly assigning treatments to our experimental units in an experimental design. So we measure a variable that we call Y, and there are many things that we may measure as researchers depending on your interest. And what is the aim of that? You just want to establish responses between different treatments so that you establish a relationship between the response and the explanatory variables. So it is about the cause and effect that you try to establish in an experiment. So we have several experimental designs, the common ones. The simplest one, usually the completely randomized design, as a CRD. And then the most used design for most field experiments is randomized complete block design. We shall look at them separately. What are the principles? When do we choose to use them? And then you have the factorial designs, which is a treatment structure that can be run in a completely randomized design or in a randomized complete block design. I'm restricting myself to complete block designs, but we have other designs we call the incomplete block designs. Like, for example, the balance incomplete block designs, the lattices, the alpha designs. These are usually mostly used by the plant breeders for large number of treatments. So the question is, if you have an experiment with 4 maize varieties, 3 nitrogen levels, you have 5 plants sown per plot. You are measuring leaf length. And then you have 3 fields, each with 12 plots. Then you have 12 combinations, which is variety by nitrogen combination assigned to each plot in the field. So the questions are, what are the experimental factors here? What are the treatments? What are the blocks? And what are the, sorry for that. What are the response for it? What are the experimental units, and what are the measurement units? So in this case, if you are to answer this, the 4 times 3 varieties are your treatments or the treatment combination. Now, the experimental factors here are nitrogen and variety. And then you have 3 fields each with 12 plots. Those are your blocks. And then you have 12 plots within each block. Those are your experimental units. And then the 5, the measurement unit are the 5 plants that are sown per plot that you're going to measure from. So that may help you to understand some of the definitions that I've given earlier at the beginning about some of the key definitions or terminologies that you need to understand in experimental units and design, rather experimental analysis and design. So then, what do we normally aim at in an experiment? The aim is basically to compare different treatments. And I will demonstrate this by looking at a completely randomized design, which is the simplest and the one that we normally use to teach this experimental design, because it is the one that you understand. Then from there, you can build on to more complex ones. So usually you have, you have a number of treatments presented by T, so it could be 5, 6, 7, whichever number. And then those treatments are allocated to experimental units at random. Now, there are some notations that you need to understand. So when I use Yij, what does it imply in real time? So this is the response of the of the ith treatment in the jth experimental unit. Then Ni is the total number of Ni is the number of experimental units receiving a particular treatment I, and then capital N represents all the experimental units. Like if your experiment is 7 treatments replicated, let's say 4 times, you'll have 28 experimental units. So in that case, NT, or the total number of experimental units in your experiment, will be 28. Now, I'm going to use an example from a book by Gomez and Gomez, Statistical Procedures for Agricultural Research. So this was an experiment on insecticides, and the response was rice yield in kilograms per hectare. There were 7 treatments, that is 6 different insecticides and a controlled insecticide treatment, with 4 replications, giving a total of 28 experimental units. So this is just an example of the randomization process. Here is simply we random across the experiment without any restrictions. We don't impose any restrictions on randomization, and this is a possible. You may have different permutations, but this is a possible layout of such an experiment. Now, this is how we use the notations. So here is a table showing the 7 insecticides under control treatment with data, raw data collected from 4 different experimental units representing each treatment. So 4 replications per treatment. So if you look at the first treatment, for example, azodrine, then the performance in all the 4 replicates are shown on top. Then the total mean for azodrine is 2,128 kilograms per hectare. So that 2,128 becomes your Y bar I dot, meaning I representing the azodrine treatment I is azodrine in that case. And then the dot means the total across all the columns. So when you use a dot or a knot is representing you're collapsing. So you're one value across all of them. So that is raw mean for that particular treatment. Now, 14,278 is the overall, means overall across the rows and the columns. So the first value on the dot is usually the row, and the second value is the column. So if I'm collapsing the column, meaning I'm looking across the column, then I'll use I representing the treatment only. Then the Yij is the single performance. The 2387 is the single performance within the group. So you'll find that a particular treatment will perform differently, but the question is why? Okay, so I is a treatment index, and then J is the group, in other words, the column value, and I is the row value. So that is the principle we use in modeling. So there are many ways of modeling. The most common one is the first one that is usually used by most people, and where you have the Yij, which is the response, individual response within the experimental unit. Then you have mu, which is the overall mean, and then tau is the effect of treatment I, and Eij, epsilon ij, is error associated with treatment I in unit J. With I, treatments from 1 up to T, and J from 1 up to J from 1 up to R. Now, the assumptions usually about the analysis of variance are usually 3 that you have to test before you subject your data to analysis. So equal variance, then you have independence of error, and then the normal distribution. These are the assumptions that we normally consider in our analysis. You'll see later, we shall look at that. Then you have a second presentation where instead of using I is equal to 1 up to T with J is equal to 1 up to R, you just say the model, and then you say with T subscript T is equal to 0. In this case, you usually set one particular treatment as your reference. So you're comparing all the other treatments with reference to your single treatment that you're using as your reference treatment. Let's say if you use the first treatment like azodrine in our case in the previous table, then the other performances are based on the differences between the other treatments in that particular treatment. And then you have the third one that is used by most of the statistical analysis. This is what you call the sum to 0 condition, where you say that the effect of all treatments across are equal to 0. In other words, if you take the effect of treatment 1, you add to the effect of the treatment 2, and up to the last, then you'll get 0. Meaning that if I take the effect of treatment 1, which is 2128 subtracted from 14278, then I add to the difference between 1316 and 1427, all the way up to 2678 minus the overall 14278. Then I will get a 0 value. So those are some of the modeling components that are applied. Usually in the model, you have 2 parts. You have the explained part, which is the overall mean and the treatment effect. And then you have the unexplained part, which is usually the epsilon ij. This is the error, is the unexplained part, because it is random. So you can't explain it. You don't know where it is coming from. Now, the estimation of parameter is such that the overall deviations of error is minimal, is what you refer to as the least square estimate or the least squares method. And here, this is what you call the point estimation. Now, when you estimate a particular mean of treatment, usually the parameter is the mean. So, and then you have the statistic, which in this case is what you estimate from your experiment, the observed value, but you have an assumed true value, which we don't know. But now when we estimate, we expect that the true value is the observed mean is equal to the true mean. So we use these observed values, like let's say y1 bar dot, to estimate the mean, the parameter value, or the parameter mean of that particular treatment, and we do for all those. Now, the standard errors of estimation we get the mean square error, which is the variance, its square root, which is the standard deviation of the estimate of error, divided by the square root of the number of observations per treatment. So you see how we do the estimation of the treatment effect is by subtracting the overall mean from the observed mean of every treatment. So with sum to zero condition is where I was saying that when you sum all those effects, then you are going to get a zero. So the common variance is the overall mean square error over the total experiment. That is the variance under the null hypothesis, which is the mean square. And you can see the formula as shown. Now, the sums of squares are usually estimated by partitioning the overall sums of square here. The overall sums of square we partition into the sums of square due to treatment, and which is the between treatment differences, and then we remove the error effect, which is the within treatment observation. Basically, we expect that the treatment should give us the same value, but why does it differ? That is where the answer lies. So the least square estimate is where the deviation, the overall deviation of the values from the true value is minimal. So the minimal error gives us the least square estimate, which is our best estimate. So the total sum in simple from these observations, remember the notations I showed where the Yij is the observation, this is the overall, and you can follow through from my earlier explanations. So you see this is the total sum of squares. If you add the errors, if we take the treatment sum of squares, we add to the error sum of squares, we are able to get the total sum of squares. So we partition the total sum of squares into explained and unexplained part of the variation with the degrees of freedom partitioned as you can see. So in an analysis of variance, our interest is basically the differences between the means, and we normally use the error, which is the within group variance, to estimate the differences, such that if the between group variance is much greater than the random variation, then we can conclude that we have significant treatment differences. So here you just see how you estimate the effects based on the model notation. So if you look at this, you can see the Yij from the raw data, the Yi dot, which is the mean of each treatment, and then the Y dot, which is the overall mean. So then you see the Yij minus the Y dot is the treatment effect, and then you have the error effect, and you can see. So if you look at the Yij minus the Y bar dot, that is the total. And then you have the Yij minus the Yij dot, that is the within. And then the Yij dot minus Y bar is the treatment effect. So this is the treatment effect here. This is the error effect. This is the total from those notations, the way they are applied. And here it is demonstrated how you come about with these values. So variation due to treatments, which is this one here, and then the within, which is the random variation, and then this is the total. So when we sum this, then they are additive, then we get 7.5, and you can see how those values are generated real time without necessarily going to the calculator to use the formula. Now, this is the ANOVA table you expect from a CRD experiment. So you have the sum of squares, you have the partition, you partition the source of variation into treatment error and total. And then you estimate the mean square, which is the variance from the sum of square error, and then you divide by the degrees of freedom. These are the expected mean squares. As you can see, this is a random variation. And so for us to estimate the F, then we divide this by that, and we can also calculate the treatment effects directly based on the error mean squares. So, like we said, that the difference is between the means, which is of our interest, which is the between group variation. So if you run the data from the example that I was showing from the book, then this is what the kind of analysis of variance output that you will expect to get. So from here, we use the random to test for the differences between the treatment, which in this case is the insecticide. So if we apply an F test by dividing the mean square error of the treatment by the within, this is the between, and you divide by the within, we get this value. So from the F values, if you look at the probabilities, we see that it's highly significant, because then based on the null hypothesis, this value, the probability is much less than the tabulated value of F. So that is how we use the random part to test the fixed part, which is the between treatment. So like here is just a demonstration that I have my treatments, and this is the trade score for which I'm measuring, and I'm just trying to see if they are different. So this is the F test based on the null hypothesis. So your null hypothesis is that all the means are equal. Now, we want to test using statistics whether that is true or whether we dismiss that. So then, based on our null hypothesis, this is similar because, you know, mu is the parameter, and then we are estimating using tau, which is the treatment effect. So based on alternative versus the null hypothesis, whether there are treatment differences, the conclusion here is that there are treatment differences based on the F probability. So how we need always, like I said, to check on the assumptions of the analysis of variance. We check on equivariance, we check on independence and normality. And how do we test that? We normally check the assumptions using our residuals. We use the half normal plots. We can also use the quantum plots to check for the normality. Now, usually you look at the symmetry. If you look at this, yes, it doesn't look normally distributed, but it is symmetrical. So if you look at this, the distribution of this, it appears to be normal. Then here, if you're looking at the quantum, then it should be more or less a straight line. It should be more or less a straight, should not deviate from more or less a straight line. Then that shows you that your data is normal. So if you are looking at the residuals, for example, you look at the midline, which is the zero line, the standardized line, then the distribution of your residuals on both sides should be distributed across. They should not be clustered. If they are clustered, then it shows you that your data is not normal. Usually you expect for normal data, most of the residuals should range between -1, -2, and +2. So when you run this kind of analysis, you can use histograms of residuals. You can use a normal plot of residuals, and you can use the quantum plot to help you to check. You can also run other analyses like the Shapiro-Wilk test, that is for normality. And if you find non-significance, then it means that your data is not deviating from normal. But if the Shapiro test is normal, is significant, that it means your data is not normal because it is deviating from normal distribution. You can also use a Tukey's test of additivity. And also, you can also use a Kolmogorov-Smirnov test. Those are some of the tests that you can use to check for all the normality tests of the assumptions of the analysis of variance before you can run the data. If your data does not meet the assumptions, then you need to transform your data, and there are different kinds of transformations you can use. You can use log transformation, you can use angular transformation, and there are other forms of transformation depending, or square root transformation, depending on the kind of data that you are dealing with. Now, H0 is, you are saying there's no difference. So should we reject the null hypothesis, because the null hypothesis says no difference? But once you test and you find that the null hypothesis is significant, then you move to the second step, which is now the multiple comparisons. And there are different kinds of multiple comparisons that are there for you to use. So you have examples, the Fisher's protected LSD. You have the Bonferroni test, you have the Tukey's test, you have the Sheffer, and several other tests that you can use. Now, they have merits and demerits, and you choose a test based on how sensitive it is in terms of helping you to get real differences rather than being wrong by committing what you call type 1 error. Because you may declare that there are differences, but it is not true that those differences exist. So this is just an example of LSD, which are just several series of tests, of t-tests that you can use. And from your basic statistics, I'm sure you know how to calculate this. But we have merits and demerits of, like, if you have an equal number of treatments, then you may use separate LSD tests, or rather t-tests. But if you have balanced data, then you can use an LSD. So you also have the Tukey's test that you can use. The Tukey test, you get these Q values from standardized tables that you can get from textbooks, and then together with your mean square error from your analysis of variance, and you can calculate, and the mean comparisons follow the same procedure like LSD. So if you can compare two tests here, this is an LSD test, so this is the Tukey's test, and this is the LSD test. So you'll get different tests because the LSD values for Fisher or Tukey usually vary. The Tukey's test LSD is usually higher than the LSD, meaning that it is not easy to declare a difference when it is not there. So it restricts you from committing the type 1 error. So the power of the test depends on how sensitive it is in terms of detecting the differences. So you don't go for a test that will give you very high type 1 error rate. You have to minimize. So in this case, a Tukey's test would be better, for example, than an LSD test. But those are debatable points in so that is basically, I went into detail about how you go about designing an experiment, the data analysis, checking for the validity of the data, and some of important key points that you need to take. So then with the next subsequent designs, I'll now not go into the details of testing for the assumptions, how do you choose which multiple comparison tests and what have you. So in this case now, I move to the next design, which is randomized complete block design. Now, this is the design that is mostly used for field experiments, and you'll see why, because a CRD experiment usually is ideal for controlled conditions. But now when you move into the field where you have no control of most of the conditions, then it becomes challenging if you use that, because it will subject you to possibility of making a lot of mistakes. So for CRD, you see, you'll just randomly allocate your treatments. For example, giving an example where you have 3 treatments and then you have 4 replicates per treatment. So then you have 12 plots. Then you'll just allocate randomly. That is for a CRD. But what is the problem with that? Now, you may end into a problem where you have with the CRD, because you're not restricting the randomization, you may find, for example, if you have a slope or a gradient, for example, and then you find that most of your treatments A are falling on the left side and the C's are falling on the right side, and then you see the B's are falling at the bottom, you see. So you have some worries there, because if you have a gradient moving in this direction, then you will not have a chance of having your treatment A, any of them appearing on the right side of the field, and that is a problem. So then that is why you introduce a design like, for example, a randomized complete block design, whereby now, based on the fertility gradient, you subdivide into homogeneous units as the role of blocking. And then now you ensure that you randomize all the treatments in every block so that each treatment has equal chance of being represented in a particular part of the field. Now the model changes because now you introduce the row J here. Now the row J takes care of the clusters of, so it helps you now to remove the error associated with the gradient that may arise if you use a CRD in this particular example. Now in this case, K is the number of times a particular treatment appears in a block. So if I say K is equal to 1, it means that each treatment will appear only once in a particular block. And then you expect no interactions. That is one of the design parameters that exist in this that helps you now to ensure that the different treatments are different, and the blocks are also different. So whichever treatment falls in each particular does not interact with any other independent. So there's no absence of interaction. So now in the ANOVA table, you're going to have the different components. You're now going to add the block component to the treatment and the sum of and the error sum of squares with the degrees of freedom as shown. So in this case now, each treatment appears exactly once in each block, and then the total number of experimental units, you just multiply the number of blocks by the number of treatments. So then it helps you now to reduce error such that what would have arisen from the gradient differences will be pulled out of the error, and that makes the error much smaller, and that is the design efficiency. So the worst you expect from RCBD is a CRD, because RCBD is more efficient in terms of reducing error. But the parameter estimation remains the same for treatment, but then now you introduce the effect of the block. So then you have the F test for the Fisher's test for the treatments, and now the Fisher's test for the blocks. The Fisher's test for the block helps you to understand whether it was necessary to use the blocks or not in your experiment. So the interest of the experimenter remains the same, comparing your treatment effects. Okay. So you test each factor separately. So now I think I've already finished. So for factorial experiment, so in a single factor, we're only looking at one factor. But now for a factorial experiment, you may have a case where you have more than one factor in the experiment, such that you are interested in looking at how those two factors interact with each other. So in a factorial experiment, you will be able to test each factor separately, or you can test both factors at the same time. So it has an advantage over the single factor experiment because it helps you to see the presence of interaction, but it also helps you like to conduct two experiments in one. What it has the component known as the hidden replication. In other words, you may need two separate experiments of equal size to be able to run those experiments separately if you were to do a single factor experiment. But of course, because you are doing a factorial, you have the other advantage of looking at both factors at the same time. How do they depend on each other when they appear together? Okay, so for example, there's an experiment. I'm just trying to demonstrate an experiment was conducted to look at the different levels of pesticides and 3 different varieties. So we have pesticide as one factor at 4 levels, and then 3 varieties of citrus fruit, the varieties being the second factor. So your response here is the yield of the fruit, but we have factor A, which is the pesticide at 4 levels, and factor B, which is the variety at 3 levels. So 8 trees were randomly selected from an orchard, and 4 pesticides assigned to the 2 trees. So the design. So this is a CRD design, a factorial treatment structure in a completely randomized design. And this is how the design looks like. So in this case, you look at, is it balanced? Balance means orthogonality or equal proportions. Is it proportionate? Like if you look at this example here is a typical design where you see a combination variety 3 with pesticide 1 is appearing twice. If you follow the colors, each is appearing twice each is replicated twice in the experiment. So it has orthogonality, means all of them are balanced. If we take pesticide variety 1 and pesticide 3, it's also appearing twice. Okay, so the unique thing about this is that you will be comparing your treatments on equal footing. All right, so when the design is not balanced, then there's a problem. We call it contamination, because you'll be using different standard errors for comparing different. So you'll not be comparing them at the same level. So just to explain, orthogonality is important. Always try to have a balance in your experiment, because it gives you a simpler or let's say more accurate results. And even how you run your experiment, you will have less work to do, but you'll also have more accurate results compared to when you have no balance, because you introduce the contamination, meaning that the error margins will not be estimated on the same footing. So if you look at this, for example, you have Nij equivalent. So you have the factor 1, factor 2 divided by the overall, so Ni dot the overall of the 2 subtracted from the individual observation of the 2. And so it is equivalent to when you just take Nij over each, and then you take the reciprocal of that, or you have the same multiplicative elements between the rows and the columns. That is what is being demonstrated. So when it is orthogonal, then the way you compare is balanced across the design. That is what is being explained here. So how do you check the balance in your experiment? If you look at the first case here, if you look at factor B and factor A combination, so you have A1 with B1, you see appearing 3 times in an experiment. This one is, everyone is appearing 3 times, so the equal number of repeats in the experiment, so there's a balance, so it is orthogonal. You just take this value, multiply by this value, divide by the overall, you get 3, meaning each is appearing 3 times, so it is balanced. Now, the second case, if you take 6, you multiply by 3, you divide by that, is not equal to 1. If it's not an even number, so it means that it is not proportionate, because you see A1 appears once, A1 B1 once, A1 B2 twice, A2 B1 twice, like that. And then, so it's not orthogonal and it is not proportionate. So this kind of factorial experiment will have less precision. Now, if you look at the third one here, if I take 9 multiplied by 6, divide by the overall, I get 2. So it is not balanced because not all treatments are appearing equal number of times, but it is proportional and it's also orthogonal, meaning that there's proportionate representation. So my estimation when I'm using the standard error to make comparisons, then there'll be a balance and there'll be orthogonality. Here, there'll be no orthogonality. So there'll be no balance in the experiment. So the model is, as you can see, this is a CRD model. So if you look at this model, the treatment, when you see a dash, that is random. This is random, this is random. So you have factor A, you have factor B, okay? The model is additive. So these are the main effects. So the model is additive. So if you see parallel lines here, it means that you have no interaction between the 2 factors unless there's a tendency of the 2 meeting. So the effects are the same irrespective of the level. So such in general, it's still an additive model, but you see there's no interaction between the effects of the 2 factor A and factor B. So the possible model now, this is 2 factors, but we haven't looked at the interaction. But now the advantage we get with the factorial, it gives us a chance now to look at the 2 together. So here is if we look at each factor separately. But here is when we look at each factor and then both factors together. So then you have the effect of factor A, effect of factor B, and the interaction between factor A and factor B. And then this is the error, the source of error, and it's also random in the experiment. Independently distributed with a mean 0 and a variance. So the ANOVA now appears, you will have the main effects of each factors and then the interaction effect, right? And then you have only one single source of error, which you are going to test for all these. But if it is a random model, if one of these factors is random, then you will have to use the AB as, but for a fixed model effect, like the one I'm trying to demonstrate here, is then you use only one random to test all of them. So R is the number of observations for each, which is the replication for each observation. So now the total sum of squares is partitioned into the both factors singly and then both factors together, which is the interaction, and then the error margin. So when it is a balanced design, like you can see, then you are going to use this error now to test for both. So the MS, you use the error term here to test for both the significance of both effects and the interaction. But like I said, if it is a random model, because I've tried to restrict myself to the fixed model, if it's a random model, if any of these factors is random, then AB becomes random and you can use it. But in this case now, we test using this single factor. Now, what is the strategy of using an F-test in a factorial? In a factorial, we peel it like I am showing here. You peel like an onion. You start with the outer, the bigger one. So if I have three factors, the first one I have to check is the three-level factor. Then I move to the two-level factor, and then I finish with the single factors. So I don't start with a single factor, looking at the single factor before I go to, because in any case, if I have an interaction, then the single factors become less meaningful, right? So then I will not start with a two-way interaction when I have a three-way interaction, and in that manner. So that is what I'm using an analogy that you peel the model like an onion in terms of layers. You start with the biggest interaction, you go down up to the single factor. Why? Because when an interaction is significant in a factorial experiment, then and then it is involving both either of those two factors, then it doesn't make much sense, because then you have to look at both together. Interaction means that the effect of one factor will affect the other factor. Okay, so factorial experiment can be looked at also in RCBD. The one I was demonstrating the fact was a CRD factorial. It was a factorial treatment structure in CRD. And now you can also look at it in RCBD. The only thing you will add now is the block component. You will add the block component, which does not exist in a CRD. And now your model is modified because you have the gamma, which is the block effect. You have the effect of factor A, effect of factor B, and their interactions. Of course, error normally and independently distributed with a zero mean and a variance. And so now this is just an example case of the analysis of variance that you're going, and the strategy that you're going to use is the same. Now, the last one I look at is the split plot design or experiment. I said for these ones, I'll not go into details because checking the assumptions, the residuals, and all that remain the same. Now, do not use a a split plot experiment when it is not difficult to allocate your experimental unit. You only use a split plot design when one of the factors is not easy to allocate to your experimental units. For example, let's say I have an irrigation experiment, and so I have high water level application and low water level application, and I'm using a sprinkler. So it will only be good to have one part of the experiment with high water on one end and the other one on the other end, so that now when I switch on the sprinkler, it is not going to interfere like I have the drought and the well-watered experiment. So then in this case, this is why you now use this kind of design where it is not easy to apply some factor, because if you apply it, it will interfere. If let's say I'm doing an animal experiment and I have a big room with cages, I would rather, and then I'm varying the lighting system. It is easier for me to have one room with high lighting and another room with low lighting, and then I put my cages inside, because it will not be easy to install low light in one cage and the next cage I have high light. Those are I'm just giving some few examples of where you can apply. So the characteristic here is that you have the main plot, which will be your larger plot, and then the subplots will be your the smaller plots. And remember, I've said it is because it is not easy to allocate one of them. So, for example, here, I'm showing an experiment where you are comparing 4 different phosphorus fertilizations in soybean with 0, 30, 60, and 120 P units, and I have 3 varieties. So it's easy to plant a single variety in a plot. And then you subdivide into subplots where now you will apply the phosphorus. So you are going to split with 3 blocks. Then you have the large plots, which will have the varieties, and then the small plots that will have the P levels. And this is a demonstration of how you're going to split your. So you're going to first of all randomize the main the whole plot first, which is the main plot. So you see you have your 3 blocks, and each variety is randomized within each block is RCBD. So, and of course you can also do split plot in a CRD depending on the conditions of your experiment. So you randomize first within the block. So you have the whole plot, and then you randomize the whole plots within. And then in the next step now, then you randomize this. You see the colors now show you the 4 levels of phosphorus that you're going to randomize within each variety. Those will be now your P levels. So then the design now, you are introducing another source of error. You see this design has 2 sources of error. And so you have always the response is random. And then you have, this is your overall mean. And then rho is the effect of the block. Then this is the effect of the variety, which is the whole plot. And then this parameter here is the whole plot error. And then you have your subplot effect here, which is the phosphorus. And then you have the interaction between the whole plot and the subplot. So this explains the parameters in your model. So there are 2 error terms here now. And this is what complicates the split plot. You have 2 error terms. You always expect that the main plot, because they are big, it has a large error term. And then this is the subplot error, which is the smaller error term. So then you have the between whole plot variation error, which is this one here. And then you have the small plot, which is your residual E. And both of them are random, meaning that you are going to test the main plot using the whole plot error, and you are going to test the subplot using the residual. Now, this is the demonstration that you're going to use the subplot residual to test the subplots, and you're going to use the whole plot error to test the whole plot effect, the differences between the varieties in this case. So you have two terms to test the hypothesis, as I explained. So you usually in this, the subplots are estimated at a higher precision compared to the whole plot. So if you are running an experiment, so the one that is of more interest should go to your subplot, and the one that is of less interest should go to the whole plot. Okay, so the advantage is just the practical aspect of it, but the disadvantage is that you lose the precision. So use this carefully when you have a difficulty in running your experiment. Otherwise, go for the factorial, because it tests at equal footing. So that is like I'm almost at the tail end of my presentation, but I just have two questions here, two sets of questions, very short ones. So the first one, what is the purpose? What purpose is served? What is it that is the purpose that is served by a good design? And then which principles do we follow to arrive at a good design? So the purpose of a good design is an efficient analysis of variance. So we need to know, to understand how our design can influence our results, our outcome. So this is the analysis of variance approach. So we always say, you'll notice as you continue to advance that ANOVA is not usually the best way, but it's the best way to think about the data and plan. Okay, we have the residual maximum likelihood. If you go further, you'll understand that the residual maximum likelihood is a better way, which I've not talked about here. It is also convenient for arranging the arithmetic. And the idea is just to partition the variation. You count your number of degrees and you identify the correct error terms that are fundamental for the second set, which is the last part now. What is the role of a statistical model? And how do we estimate parameters in the model? And the answer is the model allows us to test the research hypothesis, and the estimation of parameters and confidence intervals can be done using the least squares or the maximum likelihood estimate. And that is the end of my presentation. Thank you so much for listening.
Thank you very much, Dr. Pascal Okwiri, for this nice presentation. I think many of our participants have liked it, and it goes very deeply on the key aspects of the experimental design and data analysis. As someone said in the chat box, I have rarely seen the linear model broken down into the separate pieces, and this is a very helpful background for understanding ANOVA. So thank you very much for taking time to let us go through this. So far we have some questions, and the one in the chat box I will read, and then you
just
answer.
a minute. Let me pick a pen, please.
Okay.
Okay. Go ahead.
So there is a Kolawole Olalika who said, "Thank you for the great presentation, sir. May I ask if it is all conditions where there is no normality with log transform data? Kindly elaborate the practical approach to solving the problem of no normality in the data collected when verifying normality assumptions. And is it applicable to any experimental objectives in field datasets?"
Should I answer that?
Yes, please.
Yes, you need to check for the assumptions of an analysis of variance before you run the model, before you fit your model. And the solution is, like I showed you, those statistical tests, like the QQ plot, which is the quantum plot. You plot the residuals. Normally you plot the residuals. You can extract the residuals from your data. If you are conversant with this statistical software, you can be able to extract your residuals. Now, once you extract your residuals, you can run several tests. Now, the graphical method is what I showed. You can use the graphical method. And so you study your half normal plots. You study the QQ plot and then the random plot. So you expect that your data, your residuals should be distributed across the page with no clustering. If that happens, it means your data is normal. But if you see some clustering, or if you look at the histogram and you realize that the histogram is not symmetrical, like it is distributed on one side, then you know there's a problem. If your QQ plot doesn't fall more or less on a straight line, then it also shows you that that your data is not normal. The alternative is to run tests like the Shapiro-Wilk test. The Shapiro-Wilk test can also, if you run a Shapiro-Wilk test, if the Shapiro-Wilk test is not significant, it means your data is normal. But if the Shapiro-Wilk test is significant, it means your data is deviating from normality. So you look at the probability from the Shapiro-Wilk test. The other test is, we call it Tukey's test of additivity. This one tests for the additivity. The Shapiro is for normality. And then you have the Levine's test. The Levine test looks at the distribution of residuals, whether they are well distributed or if they are clustered. So if your data fails any of these tests, then you resort to data transformation. You do data transformation. Of course, before data, you need to check your data and look for the outliers, for example. Not all the cases that outliers are bad. You have to be sure as an experimenter that you understand your work. So it could be a value that looks strange, but is a normal value. So if you fail these tests, then, for example, if you have data that is so much varied, you have like data for you, maybe you are counting some bacteria and you find one data is probably 0 or 1, the other one is almost 100. In such a case, you may use a log transformation. Now, if you are dealing with percentages, your percentages need to fall between 30 to 70. If you have values that are below 30 and others are above 70, then you need to do a square root transformation. And you can also apply arc sign, or what you call the angular transformation. So it depends on the nature of the data. Now, after the transformation, then you have to run your data. You have to run your data again for those tests. Once it passes those tests, then you can proceed to the next step. Sometimes when the data set is too large, you may not expect it to conform, because if all of it conforms, then you may not get any variation. So if you are dealing with a very large data set, then in that case, you can run the data, but look at the error margin. So I think that is how you apply these tests.
Okay, thank you very much, Dr. Okwiri. Another question is from Levi. May you briefly unpack the concept of degrees of freedom and its significance?
Okay, the degrees of freedom help you in error estimation. So there are formulae and there are also rules that apply to the degrees of freedom. And we normally pay attention to the degrees of freedom for error in the experiment. Now, by rule of thumb, your degrees of error your degrees of freedom for error should be a minimum of 12 by rule of thumb. It should be a minimum of 12. But if you can have from your skeleton and over, but if you can have degrees of freedom of 20 and above, that is more desirable, because when you are calculating your standard errors for, let's say, mean comparison. You usually divide your mean square error by the number of replicates. So then, if you have a higher number of degrees of freedom, it means that your error margin will be reduced. And when your error margin is reduced, then it means that you'll be able to detect any small differences between your treatments. So it is important to look, if you have your degrees of freedom less than 12 from your skeleton and over, then you have to review your design. You either increase the number of replicates or you increase the number of treatments in your experiment.
Okay, thank you
Yes.
very much for the response. Another question in the chat box. Thank you very much, Doctor, for this wonderful presentation. Please touch on split plot design and when it is used.
Okay. The principle is the same to a split plot, because it means that I say that for a split plot, the driving factor is the difficulty in how you can administer or how you can assign your treatments within the experimental layout. Like I was demonstrating that like, for example, if you are doing a cultivation experiment, maybe you are using a tractor, and then you are also looking at maybe conservation agriculture. Now, it is not easy to turn a tractor in a small plot of, say, 4 by 5 meters. So you have to allocate a whole area for the tractor plow, and then you separate from the other area. So if you have a third factor, like let's say, like we had, you have cultivation method, you have variety, and you have nitrogen level. You have 3 factors. So it applies like a factorial, but now you have 3 factors where it is the hardest one here is to apply is the cultivation method, followed by the variety, then the nitrogen. So it will be better for you to actually, first of all, do the main plot, allocate randomly to the cultivation method. Then your subplot becomes the variety, which you now allocate to your cultivation method. And then the subplot becomes your nitrogen. But always be simple in your experiment. You see, if you look at the split plot, you have 2 error terms. Now, this means we have almost 4 types of standard errors, 4 different types of standard errors that we are going to use for different tests. Now, if you move to a split plot, now you have 3 error terms now. You have the error associated with the main plot, you have the error associated with the subplot, and you have the error associated with the subplot. So you have 3 sources of error and a higher series of, we can work them out, but you'll have a very high series of standard errors that you are going to use. So it complicates your experiment. You can do it, but it becomes a very complicated experiment. So we are always told that avoid complicated experiments. Even for a split plot, you are told the maximum you should go is 3 splits. But if you can do 2 splits, the better. But then it means you can still use it depending on your conditions.
Okay, thank you. Another question from Charles Peter Apunyo. Dear Doctor, thanks for the good presentation. Between 2Ks and Duncan multiple run tests, which one is best suited for agricultural experiments?
2Ks test is better in my view. Usually with statistics, sometimes is a bit objective or sometimes subjective, but objectivity is more important here. The Duncan's multiple range test is mostly nowadays discouraged by most journals. In fact, if you use DMRT, most journals will just reject your work because it is prone to the type 1 error rate. In other words, it's less restrictive. So usually how you go, like I demonstrated between Tukey and LSD, a more powerful test depends on how sensitive it is to a type 1 and type 2 error rate. So a good test should be less prone to, it should, meaning it is more sensitive. It should be less prone to the type 1 error rate. So you really don't want to fall into the trap where you are using a test where most of it is telling you that you have differences, but in the real sense, those differences are not true. So Tukey's test is more preferred in simple answer to Duncan's multiple range test. If you want to use a multiple range test, there's this test known as the Ryan Gabriel LSD test. That is the most preferred now for the multiple range test. Even SNK, which is the student Newman's Cal test, is not really preferred in the agricultural circles.
Okay, one of the latest questions before we close. Hello everyone, I'm Mensa Meshak from University of Ghana. I'm biotechnology plant. Please, I have two questions. In the area of applied science, is there a clear difference between hypotheses and objectives? And the second question, as a researcher, how can I identify that a particular experiment is statistically valid with respect to number of treatments used and the number of time treatments were replicated?
Now, a hypothesis is a research question. Now, once you come up with a research problem, they are related. All these are empirical and they are interrelated. Once you come up with a research problem, the next thing is you ask yourself some questions that you need to answer through the experiment using an experimental design. Now, the objectives are the goals that you set to achieve so that you answer to the needs that made you or drove you to conduct this particular experiment. So that question is philosophical, but they are interrelated in one sense or the other. And you remember in an experiment, usually each objective must have a question attached to it. So they are interrelated and both of them apply in one form or the other. Even if you don't directly state your hypothesis, it will come in terms of the research problem. When you are explaining your research problem, that is where you're going to show the research gaps, and that is from where you're going to raise the research questions. Now, the number of treatments depends so much on the researcher. What is your objective in the research? It is you to set the number, like when you're running an experiment, you have an objective that you want to achieve. Like, for example, if you are running a biotechnology research, mostly we are told that the high number of entries is more preferable. So I think the answer lies with what kind of experiment are you conducting? If it is like a genetic diversity kind of experiment, or you want to do a GWAS kind of experiment, by rule of thumb, in plant breeding, 200 is usually the magic number for plant breeders if you are doing those kinds of genetic experiments. But they will tell you that the statistical power will increase based on the number of entries that you have. So another point is if you are doing a genetic experiment, it will depend on the mode of pollination. You see, the cross-pollinated crops have got a smaller linkage disequilibrium block compared to the self-pollinated crops. So you usually, with self-pollinated crops, you can even have as few as, I've seen some publications with less than 100 entries. But when you are doing cross-pollinated, you expect to have probably 300 to 500 entries. But for a general experiment, because we are a mixed group here, you can even do an experiment with 4 or 5 treatments, but you look at the statistical power. So you look at the statistical power of the design, which you can test from your, when you are designing, you can test for the statistical power, which is based on type 2 error rate. So if your statistical power is 80% and above, that experiment is sound. It's very sound. So you look at the statistical power. I also told you about the minimum error degrees of freedom for the minimum degrees of freedom for error estimation also helps you to determine what is sound for your experiment. So it doesn't have a definite answer.
Okay, thank you. One last question. The first one is what is the implication of treatment degree freedom higher than error degree freedom? And the other question is a little bit more applied for a drought experiment where you have about 320 accessions and 2 irrigation levels, well watered and low stress. Which experimental design will you recommend between a split plot design and an alpha lattice design? Thank you.
Now, let me start with the second one. When you have a high number of treatments as high as 320, a lattice would be good for you. But what you need to understand, what he asked was between a lattice and?
Between lattice and split plots.
Now, a split plot is not a design per se. We call it a split plot experiment. So you can implement a split plot. You can implement a split plot in an alpha design. So your basic design is alpha, which will help you to take care of the blocks, because with alpha you can split the blocks and that will increase your precision. But then a split plot is not independent. You see, you implement a split plot in a CRD, you implement it in a RCBD, you can implement it in an alpha lattice or any other lattice design. So the answer is use an alpha, but then you can use a split plot because you have 2 factors here. So you use a split plot, but in an alpha lattice design. The first one, come again.
Yeah, the first one is
I remember if the treatment error term,
the implications of a treatment degree of freedom higher than error degree of freedom.
I think that is fatal, because you expect that the error degree of freedom should be higher than the treatment degree of freedom. Naturally, that is what you expect. The implication is that you may have a very small error term if your degrees of freedom are very low, And the implication is that most likely your test for the significance will fail. Because if the degrees of freedom for error are low, basically you expect to have a small sigma square, the small error mean square. And that will imply that when you come to the test, then you'll not be able to see the differences, because the random variation may end up being larger than the treatment variation.
Okay, thank you very much, Dr. Pascal Ojwang, for this, for taking time to let us understand more about the experimental designs and data analysis. I think we appreciate your time and the effort you put together to be here with us. Few announcements before we close. Let me share my screen. So please help us improve our next webinar by submitting your feedback using the QR code. And ensure you follow us on our social media platforms to be informed of our next events. So the LinkedIn is African Plant Science Symposium series, and on the YouTube channel you can have all the records of our past webinars and the next ones. For those asking for the slides, please be ensured that we will share the recordings once we are done with the webinar. So feel free to share with us your feedback. Otherwise, thank you very much for all of you for attending this session, and have a good evening, morning, depending on where you are following us from. Thank you very much.
Okay. Bye-bye.
Yes, goodbye, Prof.
Thank you very much, Prof.
You're welcome.
Bye. Thank you very much, Prof.
Thank you very much. Bye.
Bye.
Thank you very much. Bye.
Bye-bye.
Thank you very much. Bye.
Yes, thank you very much. Goodbye.
Bye-bye. Bye-bye.
Make sure you send us your feedback, please. It will help us improve our next events.
Doctor, thank you.